Okay, welcome to the Protein Synthesis Mastery. What you see in front of us is a model that we'll be using in class, composed of a model of DNA, which you can see is a double-sided molecule with the phosphates being our black circles and our white pentagons being our deoxyribose sugars, our colored pieces being our nitrogen bases. So we have two sides, phosphate sugar backbone, and then our nitrogen bases that match up in the middle, A with T, G with C. The second part of the model you'll see is an mRNA. mRNA again has the sugar phosphate backbone, this time with a different color sugar, which is going to be brown in our model, which is the ribose sugar, and then the colored nitrogen bases as well. Notice that mRNA is single helix. Your nitrogen base that's pink on our models is going to be uracil, which takes the place of thymine in DNA. The other pieces of the puzzle, or the other pieces of your model are our tRNAs. You'll notice that the tRNAs are 3 nucleotides long with a holder on the back. The holder is going to hold the amino acid that each of the tRNAs individually carry. So the purple pieces that I'm putting down on top of the model right now are the amino acids you'll be using in our mastery. And finally, the ribosome. It is the cellular factory that produces protein. Protein synthesis happens in 2 distinct stages. Stage 1 being transcription, stage 2 being translation. Ultimately, we would like to read the gene and code for a protein that ends up with a trait. Transcription happens inside the nucleus. So here's our nucleus. We can imagine our nucleus being here. And here's our DNA. Our DNA obviously is much bigger than our model, but we can imagine our DNA being millions of base pairs long. Transcription starts by the DNA unzipping right at the gene. Now, our model comes all apart, but really we've seen this big long line of DNA, and we're just opening up a small little bubble inside of our DNA where that gene is. As that opens up, the mRNA nucleotides, which you'll see here as being a phosphate, ribose sugar, and a base, start to match up against the DNA as it opens up. They base pair much like DNA does, G with C, but instead this time will be A with U. The nucleotides match up, and each match up individually, and as they match up individually, the bonds are being created to create the full mRNA. Once it gets to the end, and we have the full mRNA, the mRNA is then released, and the DNA zips back up. The mRNA hangs out in the nucleus. So from transcription, we go into translation. Translation starts when the mRNA leaves the nucleus and goes out into the cytoplasm. In the cytoplasm, a ribosome will attach to the first codon of mRNA. Codon is just simply a sequence of 3 bases. This one happens to be U, A, U. The ribosome is going to read the codon and send out for a tRNA with the opposite or the matching or the anti-codon. And this one will match up right here. You can see the A's match up with the U's in this one. It's carrying an amino acid, and it's going to attach the amino acid to the ribosome. And then the tRNA will leave as it gets kicked out when the ribosome moves to the next codon. The ribosome will read the next codon, C, G, A, and send out for the anti-codon for that one. The tRNA comes with that anti-codon. It's also carrying its specific amino acid, and it will also attach its amino acid to the one that's already there, and then get kicked out. And when they get kicked out, they'll go back into the cytoplasm where they'll find their amino acids. Moves down to the next codon. The tRNA comes in the next codon, matches up, and attaches its amino acid to the one that's already there. It will do this until it reaches the end of the mRNA. So now we have a chain of amino acids attached to our ribosome. This chain of amino acids will be released into the cytoplasm. It will fold and twist and turn, and it will become a protein. That protein will then control whatever trait the gene was coding for in the DNA. Now that gene could be brown eyes, it could be brown hair, it could be skin color, it could be something more complex like an enzyme to control for digestion of a certain molecule. Once that is released, the ribosome will release the mRNA, which will, then the mRNA will then go into the nucleus and break back up into its nucleotides, where they will then be reused when the DNA opens up a next gene. And that ends translation. Now, if we compare the DNA to the mRNA, we see some differences. One difference is the simple fact that we have two sides of DNA, which we'll call a double helix, and this mRNA only has one. The one will be a single helix. So we have a double helix structure for DNA, single helix structure for mRNA. You will see that we use different sugars. In DNA we have deoxyribose sugar, and in mRNA we have ribose sugar. The base thymine in DNA is replaced by the base uracil in mRNA, so that is another difference. And the final difference that we see in between the two will be where they're located. The DNA will always be located in the nucleus and will not be able to leave the nucleus. The mRNA, however, will start off in the nucleus, but also will end up outside the nucleus and will have the ability to move back and forth. So that is the fourth difference.